Okay, good afternoon, everyone. For the last time this semester, welcome to lecture 59 of BMS 300. All right, so today we are going to go through the PollEverywhere questions for the reproductive system, and I'm sure you had a chance to see the questions ahead of time, and so I'll post the question, and then you can select the answer, and then we can discuss which the correct answer is. And then once we are done with those questions, I want to just give you a bird's eye view of what we've been discussing for this entire semester, right? And it's the same slide I showed you on the very first day of lecture, right? And we'll see how everything makes sense to you now that you've had a chance to go through the entire semester. So here's question number 1. There it is. All right. Which of the following statements are true, right? Is it the anti-Mullerian hormone is necessary for the development of the Mullerian duct, or is it that estrogen is necessary for the development of an ovary, or is testosterone necessary for the development of testes, or is chromosomal sex determined, or does chromosomal sex determine the gonadal sex, which then will determine the phenotypic sex? So which of these statements do you think would be true? Is it locked? There we are. Okay, so most of you picked option D, and option D is correct, right? And we see that it is the chromosomal sex which determines whether you have testes or ovaries. So it will determine which gonads are formed, and then depending on the gonads, there will be various hormones which are produced, which will in turn determine the masculinization or the feminization of the external genitalia and also other parts of the body as well. But we see that for the development of the Mullerian duct, right, you don't need any hormones at all. It will just by default develop. You don't need, in fact, the anti-Mullerian hormone will cause the regression of the Mullerian duct, and neither estrogen or testosterone is necessary for the development of ovary or testes. As shown in this figure here, it is the chromosomes which will determine whether ovary or testes are formed. So is this clear, folks? Why option D is the only true statement among these four. All right. So an XY individual with androgen insensitivity disorder would develop which of the following? Would this individual have ovary and Wolffian ducts? Would there be testes and Mullerian ducts? Would there be ovary and both Mullerian and Wolffian ducts? Or do you think there will be testes with no ducts at all? So this androgen insensitivity disorder could be due to the mutations in the receptors for androgens, right? And that could be the reason why, even though there may be high levels of androgens, they cannot function. Okay, most of you picked option D, testes and no ducts, and you are correct, right? And the reason is that the presence of this Y chromosome is going to lead to the formation of testes, as shown in this figure here. So the Y chromosome has the SRY gene, which in turn will produce the SRY transcription factor, which will upregulate the SOX9 transcription factor, and that will cause the suppression of the expression of these genetic factors. And as a result, there will be testes formed. And we know the testes will produce two types of hormones. There will be the testosterone and the anti-Mullerian hormone. And the anti-Mullerian hormone will cause the Mullerian duct to regress, right? Whereas testosterone is meant to maintain the Wolffian duct, but that cannot happen because the receptors for testosterone are defective, right? And that's why there would be testes, but there would be no ducts. Is this clear, folks? Yes? Why a person with an XY individual with androgen insensitivity syndrome would have no ducts, although that person would have testes. Okay. RSPO Wnt4 is responsible for which of the following? Is it for initiating puberty, triggering ovulation, development of an ovary, or maintenance of the corpus luteum? Which of the following do you think RSPO1 and Wnt4 are responsible for? Okay. So most of you picked development of the ovary, and that is correct, right? So as I mentioned in the previous slide, we see that these three genetic factors, RSPO1, beta-catenin, and Wnt4 signaling molecules, they will lead to the development of the ovary, right? And whereas initiation of puberty and then triggering ovulation and the maintenance of a corpus luteum, the other options, they're all regulated by hormones, right? Whereas this is by genetic factors. Okay, moving on. After completion of meiosis II, all gametes are diploid, haploid, aneuploid, or euploid. So what do you think would be the condition of the gametes after meiosis II is completed? Okay, most of you picked haploid, and that is correct. So we see that in this slide it's showing you mitosis and meiosis. So meiosis is on the right hand panel, and at the end of meiosis you have only one copy of the chromosomes, and so it is said to be haploid, right? Whereas at the end of mitosis you have an identical daughter, two identical daughter copies of the parent cell, and they are diploid with two copies of the chromosome. Yes?
So in the egg, though,
Mm-hmm.
meiosis II is halted until the sperm actually penetrates into the egg. So
Right.
then it would release the second, was it polar body?
Yes?
But then it would still be diploid because the sperm
Mm-hmm.
haploid genes are already in there, right?
Yeah, but that is after fertilization. But this is just asking after meiosis II is complete. I see what you mean, right? But this is just asking what happens at the end of meiosis II. So yeah, I should maybe say male gametes. Would you be happy with that answer then? Okay, great. Yes, let's make it male gametes. It's great when you have students finding correcting some things in the slides. That means there's hope for the future. That's what my mentor said. This was back at Washington State when someone caught something in the slides. He said, okay, there's hope for the future now. All right. So moving on. Thank you for that. A mutated Y chromosome that lacked an SRY gene would lead to which of the following? Would it lead to the degradation of the Wolffian duct, or would there be development of testes, or activation of the SOX9 gene, or would there be masculinization of the external genitalia? So if the Y chromosome does not have the SRY gene, which of the following would occur? Is it locked again? Okay. There we are. Okay, most of you picked degradation of the Wolffian duct, and that is correct. Okay, so we see that as shown in the slide, in order for the Wolffian duct to not be degraded, there has to be testosterone produced, right? That's why I said the wolf needs to eat testosterone in order to survive. And so we find that if the SRY gene is not present, then you don't have activation of SOX9, right? And if you don't have activation of SOX9, then we will have the ovary being produced, and if the ovary is produced, you won't have testes, and so you won't have testes producing testosterone, and as a result, it would lead to the degradation of the Wolffian duct. Whereas the SRY gene not being present won't lead to the development of the testes or the activation of SOX9 or the masculinization of the external genitalia. Does it make sense, folks? Yes? Okay, moving on. Which of the following does not develop from the Mullerian duct? Is it the uterus, the uterine tubes, also known as the fallopian tubes, or the oviducts, or would there not be development of the cervix or the lower part of the vagina? So which of these following do not develop from the Mullerian duct? Okay, most of you picked the lower part of the vagina, and that is correct, right? And the reason is that in this slide I showed you that it is the uterine tubes, and then the cervix and the uterus, all of that, and also the upper part of the vagina would develop from the Mullerian duct, but the lower part of the vagina would be part of the external genitalia, and that development is going to not be from the Mullerian duct. Okay, moving on. In males, you would find receptors for the follicle stimulating hormone where? Would it be in the granulosa cells? Is it in the nerve cells, the theca cells, or the interstitial endocrine cells, also known as the Leydig cells? Which of these things would you find the receptors for the follicle stimulating hormone in males? Okay, most of you picked nerve cells, right? But I see that most of you did not pick granulosa cells or theca cells, right? And option A and C, granulosa cells and theca cells, they are found in the ovaries, not in the testes, right? So that's why both those two are not right options. Whereas we find that it is the Sertoli cells or the nerve cells which will contain receptors for follicle stimulating hormone, because as shown in this picture here, notice FSH will act on the nerve cells to have various effects. Does it make sense, folks? Yes? All right. The path of the sperm through the male reproductive system is blank. So do you think the testes, from the testes, where would it flow through? Would it flow through the path given in option A, B, C, or D? It's a straightforward anatomy question. If you just know where the sperm flows from. Okay, most of you picked B, and that is correct, because as shown in this picture here, notice that the sperm will be produced in the testes, and from there it will move into the epididymis. So option B says epididymis. From there, the epididymis will give rise to the vas deferens or the ductus deferens, which will then loop over to form the ejaculatory duct, right? And there's no ejaculatory duct mentioned in option B, but from there it would move to the urethra, where it will eventually be sent out of the body. So the answer is option B. Okay, fibrinogen forms a temporary clot in the vagina, right? And which of the following secretions produces fibrinogen? Is it the seminal vesicles, prostate gland, or bulbourethral gland, or does the epididymis produce the fibrinogen, which will cause a temporary clot in the vagina? Okay, fantastic. So most of you said seminal vesicles, and the right answer is shown in this slide, right? So notice that the fibrinogen forms a temporary clot in the vagina, and this is one of the components of the seminal fluid, right? And I hope you know what are the various components of the various secretions by the accessory glands. Is this clear for us? Yes. Yes, seminal vesicles and gland, the term is used interchangeably. Yeah. Anything else, folks? Any other questions? Okay, moving on. The blood-testis barrier is located between which and which? Is it between the capillary endothelium and the seminiferous tubule, or is it between the primary spermatocytes and the secondary spermatocyte, or is it between the spermatogonia and the primary spermatocytes, or is it between the secondary spermatocytes and the spermatids? So where would you find the blood testis barrier? Most of you picked option A, the capillary endothelium and the seminiferous tubule, but that is not the right answer, right? The right answer is option C, between the spermatogonia and the primary spermatocytes. So if you look at this figure here, notice that the blood testis barrier is denoted by these dotted lines, and these dotted lines are indicating the tight junctions which are formed between the nurse cells. So these nurse cells are shown in purple. They're these really large cells, and there is the tight junction between these cells. Now if you notice here that towards the basal compartment of these nurse cells, you have the spermatogonium, and that spermatogonium, once it undergoes mitosis and forms the primary spermatocyte, the blood testis barrier will give way, like kind of like sliding doors, for it to move into the lumen of the adjacent nurse cells. And then the primary spermatocyte will undergo meiosis to form the secondary spermatocyte, and then eventually it will form the spermatids. But the blood testis barrier is separating the primary spermatocyte from the spermatogonium. That's where it is found in the seminiferous tubules. Is clear, folks? Yes. Okay. So sperm entering the lumen of the seminiferous tubules, are they mature but immotile? Are they mature and motile? Are they immature and immotile? Or are they immature but motile? What do you think would be the properties of the sperm which is leaving the seminiferous tubule? Okay, most of you say mature but immotile, but notice here that the sperm is produced in the seminiferous tubules, and then it moves into the epididymis where maturation takes place, right? So maturation takes place in the epididymis. So the right answer would be C. They are immature and they are immotile. In order for motility to happen, they have to mix with the fluids from the seminal glands. Does it make sense, folks? Why the answer is option C? Okay, great. The final step of capacitation occurs where? Is it in the tail of the epididymis? Is it in the ductus deferens or the vas deferens? Is it in the urethra? Or is it in the female reproductive tract? Where does the final step of capacitation take place? So we talked about what are the various changes which need to take place in the sperm before it can finally fertilize the egg, right? And so we talked about where capacitation takes place. Okay, most of you said the female reproductive tract, and that is correct. So notice here that there are some proteins which are being produced in the testes, in the head of the sperm, and then there are more proteins produced as it goes from into the epididymis, and then as it's ejaculated there are other proteins being produced. But finally we find that when the sperm is capacitated, that there is going to be rearrangement of the proteins in the head of the sperm, and this last step will take place in the female reproductive tract because of the secretions of the PEG cells which are found in the fallopian tube. So if I were to modify this question, right, and I ask you, where does the first step of capacitation take place? In these four options, which do you think would be the best answer? So I hope you're able to see how a small alteration in the question, if I change the word final to first, what do you think the answer would be? How many of you think it's A? B, C can be D, right? So I would say the answer is C, right? And let me explain why. So we find that the first step of capacitation is where the sperm comes in contact with the secretions of the seminal fluid, right, which will make the sperm motile, right? And we find that the tail of the epididymis is a place where the seminal fluid will not come in contact with. Even the ductus deferens, it really won't come in contact with, because the ductus deferens will turn into the ejaculatory duct, and then secretions from the seminal vesicles will flow into the urethra. So I would pick the answer as urethra, where in these options the place where the first step of capacitation would take place in the urethra. Does that make sense, folks? And if there's any clarification which is needed, I would just take you back to this slide. So notice here that the seminal vesicles or the seminal glands is going to release its secretion, right? It's going to release into the ejaculatory duct, which will eventually join with the urethra, right? So that's where the first step of capacitation would take place. It won't take place in the epididymis or in the vas deferens. Does this make sense? Okay. Yes.
Would it occur in the seminal?
But that wasn't an option, right? So it would happen in the ejaculatory duct, because that's where the secretions from the seminal glands enter in. But if ejaculatory duct was an option, that would be an answer. But if urethra was given, that's also a viable option as well. Okay, so we now move on to the next question. The female germ cells will enter meiosis 1 before birth, during puberty, at fertilization, or during menopause? Yes? During puberty? Okay. All right, let's see what the majority of the class says. So most people said before birth, and that is correct, right? And here's the reason why. We find that initially the oocytes, they're going to increase dramatically in number, right? Because it's going to undergo mitosis, right? But then the mitosis is going to be arrested in the after mitosis, and it enters into meiosis 1. It'll be arrested after prophase 1, right? So that's going to happen before birth. So that's why the answer is the female germ cells will enter meiosis 1 before birth, then they'll be arrested at prophase 1, and then the baby will be born. Is this clear, folks? This question is based on this flowchart here. Okay, moving on. The female germ cells will complete meiosis 2 when? Will they complete meiosis 2 before birth, during puberty, at fertilization, or during menopause? Can be option A, right? Because we talked about it entering meiosis 1 before birth. Okay, most of you picked C, at fertilization. Yes, that is when meiosis 2 would complete. And again, I want to remind you of what we saw in the video, right? We find that when the ovulation takes place and the oocyte is released, it's going to be released as a secondary oocyte which has started meiosis 2, but it's going to be arrested at metaphase 2. That's when the oocyte will be released. And finally, when fertilization takes place, that's going to trigger mechanisms for the meiosis 2 to complete, right? So meiosis 2 completion takes place at fertilization. Does this make sense, folks? Yes? Okay. A large surge of luteinizing hormone secretion is a trigger for blank. Is it for the onset of puberty, or ovulation, or menstruation, or for menopause? What would happen when you have a surge in the levels of luteinizing hormone? Okay, most of you said ovulation, and that is correct. So if you notice this figure here, notice that the follicles are being developed during the secretion of the follicle stimulating hormone, right? Whereas when the luteinizing hormone is released, there's a high surge of luteinizing hormone, ovulation takes place where the follicle will rupture and the oocyte is going to be released into the female reproductive tract. Does it make sense? It's a pretty straightforward question, just knowing what happens when the various hormones are released. Okay, which uterine phase is marked by the initial buildup of the endometrium in response to rising estrogen levels? So the uterine lining is going to undergo changes during the monthly menstrual cycle. So what phase do you think will happen during the buildup of the endometrium? Is it the secretory phase, proliferative phase, luteal phase, or menstruation? Which do you think will be activated by rising estrogen levels? Okay, most of you said proliferative phase, and that is correct, right? So again, coming back to this figure, notice that as the estrogen levels are going up, if you look down at the uterine lining, you have the proliferative phase where the cells in the endometrial lining are undergoing mitosis and they are dividing, and there is the growth of the endometrial lining. Whereas when you have the levels of progesterone increasing, that's going to maintain the endometrial lining, right? And eventually, as the progesterone level drops off, the maintenance of the endometrial lining can no longer continue, and that leads to the menstrual phase where the endometrial lining is shed. Is this clear, folks? Pretty straightforward on, again, what hormones are causing which type of changes in the endometrial lining and also in the ovaries as well. Okay, high levels of GnRH, also known as the gonadotrophin releasing hormone, will lead to an increase in the follicle stimulating hormone from the anterior pituitary gland, or will there be testosterone released from the theca cells, or will there be inhibin released from the granulosa cells, or do you think all of the above can happen when you have high levels of GnRH being released? Okay, most of you said all of the above, and you are correct, right? So if you notice these two upper panels, it will clearly show you that when you have high levels of GnRH, it will lead to the release of follicle stimulating hormones. So option A is right. And then if you look at this lower panel here, we will see that even androgens like testosterone will be released from the theca cells in response to GnRH release, which in turn will release luteinizing hormone, which will cause the androgen formation. And similarly, inhibin is also produced by the granulosa cells in response to high level of follicle stimulating hormone, which was released due to the high levels of the GnRH levels. So is this clear why options A, B, and C are all correct? Yes? Okay. So we see, moving on to the next question, high levels of estrogen from the dominant follicle causes a blank feedback to the hypothalamus that leads to blank levels of luteinizing hormone. So will there be a positive or negative feedback, and will it lead to either an increase or decrease in the levels of the luteinizing hormone? What do you think will happen when there are high levels of estrogen? Okay, so most of the class said option A, but the answer is actually option D, right? Let me explain. So there will be a positive feedback to cause an increase in the levels of luteinizing hormone. So to explain this answer, I need to show you what's happening in this figure here. So in this region here, there are these two ovals, right, and they're showing you what happens with changing levels of estrogen. So early on, we find that the estrogen levels until day 10 aren't high enough, and so the low levels of estrogen will lead to the inhibition of the luteinizing hormone secretion. So notice here that this red line is showing you how the luteinizing hormone secretion is inhibited because the estrogen levels aren't high enough. But then after day 10, the estrogen levels will reach a high enough level by day 14, where it leads to the release of the luteinizing hormone, which will in turn lead to ovulation. And so it makes sense that the luteinizing hormone isn't released until the oocyte is sufficiently developed, right? So that's why high levels of estrogen from the dominant follicle will lead to the release of the luteinizing hormone. Again, if this question was changed from high to low levels of estrogen, what do you think the answer will be? It would be negative feedback leading to a decreased secretion of luteinizing hormone. Is this clear, folks? Questions are like this are great. All I do is change one small thing and I get a new question. So moving on. Labor involves which of the following? Will there be an increased progesterone to prevent the degradation of the corpus luteum, or will there be an increased relaxin to inhibit oxytocin release, or will there be increased oxytocin to promote myometrial contractions, or will there be the contraction of the cervix? Which do you think will happen during labor? Okay, we have 85 responses. Okay, most of you picked option C, the increased oxytocin will promote myometrial contractions, and you are correct. So we see that oxytocin is going to cause the contraction of the uterine muscles, which are in the myometrium, and that is going to cause the fetus to be pushed downward, and that will cause the dilation of the cervix, right? And as the cervix dilates, that's when the baby can be delivered, right? Whereas contraction of the cervix won't allow the baby to be delivered. So the right answer is C. Does it make sense? Yes? Okay, moving on. During the first trimester of pregnancy, the corpus luteum is prevented from degrading by which of the following? Is it by the HCG secreted by the placenta, prolactin secreted by the anterior pituitary gland, estrogen secreted by the follicle, inhibin secreted by the granulosa cells? So which do you think will prevent the corpus luteum from degrading? Okay, most of you picked A, the human chorionic gonadotrophin released by the placenta, and that is correct, right? So when I went through this slide, I told you that HCG is is the indicator of pregnancy, and this will be released by the placenta to prevent the degradation of the corpus luteum, so that there will be sufficient levels of progesterone maintained until the placenta is well developed enough to produce its own progesterone, right? That's why HCG is the one which prevents the corpus luteum from degrading. Okay, infant suckling stimulates the secretion of blank to produce milk and blank to eject milk. So what do you think the infant suckling would lead to? Which hormone produces milk and which hormone leads to the ejection of milk? Okay, Okay, most of you picked prolactin and oxytocin, and you are right. So we see that the suckling is going to send signals to the brain, right? And so in the hypothalamus, you will have the production of oxytocin, which will lead to the milk letdown, whereas prolactin will be produced in the anterior pituitary gland. So anterior pituitary gland will produce prolactin that will act on the mammary glands to produce milk, whereas oxytocin will act on the cells in the mammary gland to contract and to squeeze out the milk. So the answer is option D. Okay, which of the following acts as a contraceptive by directly inhibiting a hypothalamic hormone? Is it follicle stimulating hormone, estrogen, progesterone, luteinizing hormone? Bonus question, what is the name of this hypothalamic hormone? Okay, most of you picked progesterone. Progesterone is correct. So what is progesterone inhibiting? The GnRH, right? The gonadotrophin releasing hormone shown right here. And without the GnRH being released, you cannot have FSH, which is necessary for the development of the follicle, and also luteinizing hormone, which will cause the ovulation, where the egg is released from the follicle. Okay, moving on. Which of the following STDs is caused by a bacteria? Is it trichomoniasis, syphilis, genital HPV, or AIDS? So which of the following is caused by a bacteria? Okay, most of you picked syphilis, and that is correct. So it's caused by Treponema pallidum. That is the name of the bacteria which causes syphilis. And I hope you know the name of what pathogen is causing these 6 kinds of sexually transmitted diseases. Okay, the nausea and the tiredness during the first trimester can be attributed to the effects of blank. Is it due to the effect of the human placental lactogen, estrogen, progesterone, or is it the human chorionic gonadotrophin? Which of the following will lead to nausea and tiredness? Okay, most of you picked HCG, human chorionic gonadotrophin, and the answer is given right here, right? It is this hormone which leads to nausea and tiredness as well. And this is a way of the placenta letting the mother know that you are pregnant, right? Something is happening, so please take care, because the first trimester is the time when the fetus is most vulnerable. Okay, here's a question where we're tying the clinical manifestations due to pregnancy to various mechanisms. So the swelling of ankles and face during pregnancy is due to blank. Is it due to an impact of the hormones on the connective tissue, increased blood volume, reduced insulin sensitivity, or is it due to the activation of the melanocyte stimulating hormone? Okay, most of you picked option B, increased blood volume, and that is correct. So we see that as pregnancy progresses, the blood plasma is going to increase by 30 to 50%, right? And that's why there will be more fluid collecting in the interstitial fluid, and that will lead to swelling of the extremities like the hands and face and so on. So now we come to this slide where we look at the overall integration of the various concepts we discussed, and we end the semester by talking about fertilization, which leads to the formation of a cell, which can eventually form the entire human being. But we find that the semester was started by looking at a structure of a single cell, and the major component of a single cell is water. right? So this led us to the discussion of the properties of water, or the properties of water and how various substances will interact with water. And so we see that some are hydrophilic, which means they are water loving, and others are hydrophobic, which means water hating, and others have amphipathic properties, which means one part is hydrophobic and the other part is hydrophilic. And so we find that the cell membrane is made up of these amphipathic molecules, which are arranged in this phospholipid bilayer, and this phospholipid bilayer is going to not let anything get in or out of the cell, especially if it is hydrophilic. And so the regulation of various molecules moving in and out of the cells is possible through these channels made up of transmembrane proteins. And this led to the discussion of how proteins are synthesized, and we saw that that the nucleus is where the genetic material is, and based on the genetic material, the genetic information, you have the central dogma explaining how the proteins are formed. And we saw how the proteins, once they are formed, they can be acted on by various organelles to ensure that they have the final structure for influencing the specific functions. So one of the types of proteins which we looked at in greater details are proteins which help hold cells together, and that led to the discussion of cell junctions. So we have these tight junctions which can form a near leak-proof seal between the cells, and I told you that these are like fences around a house, right? And the presence of these tight junctions means that the entry into the house can be only through a certain way, right? Just like how traffic is regulated outside Clark Hall because of the construction, we find that tight junctions can regulate traffic. We also have these gap junctions which connect cells, so that from one cytoplasm there can be ions, molecules, nutrients flowing from one cell to the other. And there are also these anchoring junctions which offer mechanical support, especially to parts of the body where they are under extreme stress, like the cardiac muscle and also the skin. And then we talked about how groups of cells work together to form different types of tissues, and we considered the role of the nervous tissue, muscular tissue, epithelial, and the connective tissue. And pretty much every organ system we considered is got these tissues, and we saw how they functioned. And we found that the energy required for making proteins or for pretty much any other cellular function comes from ATP, which is the energy currency in the body. And ATP can be made from macronutrients such as proteins, carbohydrates, and fats, which are present in the bloodstream due to the functioning of the digestive system. And for the digestive system to function optimally, we see that the autonomic nervous system plays a role where you need the regulation by the parasympathetic activation, and there is also the voluntary functions like chewing and swallowing, which also the central nervous system will control. And we find that the endocrine function is found in the digestive system organs, specifically in relation to the pancreas, which releases a number of hormones. We also find that if there is a drop in blood glucose levels, the adrenal glands can get involved, where you have the adrenal cortex and the adrenal medulla releasing hormones to ensure the blood glucose level is maintained. And we found that the hypothalamic pituitary axis will help produce hormones which can have an impact on various parts of the body. And an example of the organs affected by hormones released by the hypothalamic pituitary axis are the gonads, and we saw this in quite a bit of detail in these last few lectures. Now we find that the reproductive system is connected to skeletal function as well, because in women after menopause there is a drop in the estrogen levels, which can lead to a decrease in the bone density. So while we're on the topic of bone density, we see that we can increase bone density, especially when you're young, when your osteoblasts are more active than the osteoclasts. That's when you increase bone density, and that's by maintaining a proper diet and also by engaging in weight-bearing exercises. So to do weight-bearing exercises, you need the skeletal muscles to move, and we find that if you are, let's say, running away from something dangerous, again the movement of those muscles will be influenced by the fight or flight mechanism. So this is the regulation by the sympathetic activation of the autonomic nervous system. So while you have the fight or flight response being activated, it will lead to changes in various parts of the body, including an increase in the rate of in the heart rate, because blood needs to be pumped at a faster rate to the various parts of the body, and the blood composition will include nutrients being carried to various parts of the body, and those nutrients come in through the function of the digestive system. And then the blood will also carry gases which are obtained by the respiratory system. When you inhale in air, oxygen is taken to the various tissues, and carbon dioxide can be taken from the tissues to the lungs where they are exhaled out. Now the blood can also be filtered by the urinary system, and it helps to maintain the homeostatic balance in the blood. And the blood also has a number of immune cells as well. And some of these immune cells are also found under the skin, so as under the integumentary system. And if there is, let's say, a splinter which were to get into your skin, that will activate the innate immune response, which can in turn play a role in activating the adaptive immune response. So that's how all of these concepts are connected. So before I end, I want to thank my teaching team, especially Michelle Soros, our course coordinator, and I also want to thank Kaylee and the undergraduate teaching assistants and other GTAs like Ebony, Hannah, Ian, Kiana, Colton, Kendry, and Kristen. Right. So before you guys leave, I hope there's at least one thing you take away from this course, and that is having a sense of gratitude. Because we talked about a number of pathologies in this course, and I gave you examples of what can potentially go wrong, and if you don't suffer from at least a few of those, I think there's a good reason to be thankful, right? So thank you all for your time, and I hope you had a great semester, and I wish you all the best for the future. Thanks.